Hello, it's Dr. Ross. Welcome back to the last week of responsible and ethical conduct of research training for Biology 104 this semester. We're going to tackle the last subject this week, the concepts of research negligence versus research misconduct and fraud. So this is my favorite topic, primarily because I think it's, well, these are all applied to our class and to your lives as scientists. But I think this is also the one that's fraught with the most peril because there are a lot of norms to understand about what not to do to ensure that you don't get accused of research misconduct, which is the same thing. It's a synonym for research fraud. And as we've done in the past, we have a shared Google Doc for you to take notes on and participate in some asynchronous discussion. So please open a navigator window, a web browser, go to tinyurl.com//F, numeral 2, numeral 1, lowercase, RECR, numeral 6. That's tinyurl.com/F21, RECR 6. This is week 6, our last week, and you do need to be logged into your Fresno State Gmail account to be able to access that document. So let's get into negligence versus fraud. So this falls under the broad category of research misconduct. This is critical for you to understand as a growing scientist and as a student in this class, especially since you're going to be working on research projects. So what we want to do here is to make sure that you don't get in trouble like this person who's being pulled over by the police officer in the image in the bottom right. We don't want you to get in trouble because you don't know what the norms and the rules are for how to maintain and deal with data. So initially, the definition of research misconduct involved these words that you can see here. We've already talked about plagiarism. We've already talked about human subjects protection, but fabrication and falsification, making up data, inventing data, or editing or altering your data. That's the fabrication and falsification parts. That's really what we're here to talk about today. And then the difference between negligence and misconduct and fraud has to do with this bottom point. That negligence is what happens if you break a rule and you didn't know it was a rule, right? So it's sort of an innocent fault that you did something, but you didn't know it was illegal. You still get pulled over for it, right? But it's worse if you conduct misconduct or fraud, which is where you knew the rules and you chose to break them, usually for your own gain, right? To get a nice looking chart for a paper, to get your paper published, whatever. So here is, for your use, again, another academic policy manual from Fresno State, APM 510. I'm going to focus on that box there. So this is our campus definition of research misconduct. And I'm going to place emphasis on one particular part of falsification. So fabrication, I already said, part A here, making up data. Of course, you cannot do that. That's not allowed. Falsification is important to focus on here. Manipulating research materials, equipment, or processes, or changing or omitting data, with emphasis mine on omitting, leaving stuff out, such that the research is not accurately represented in the research record. This is another reason that that record keeping module we talked about a couple of weeks ago is important. Accurate representation of the data is critical so that you don't get accused of research misconduct. But now, today, we're going to talk about what not to do that actually counts as changing or omitting data or results. So today's focus is on data and what to do and what not to do with data to avoid being accused of misconduct. And because I happen to love this particular aspect of data management, I'm going to focus today's conversation on image manipulation. Okay, that is not just like recording numbers in your lab notebook, but when the data come as an image, what are the things that you can and can't do with images that will help you avoid being accused of either negligence or misconduct? So here's an example. There's something wrong with this image that scientists spotted. It's really subtle, that suggested to them that this image had been manipulated. So see if you can spot it. So here's what somebody found that made them suspect that this image had been doctored. And if we zoom in, it's really subtle, but you'll see that between two lanes on this agarose gel, which I'm circling here with the laser pointer, there's a sort of striking difference. It's a straight vertical line where the shading of the gray on either side doesn't match up. And that suggests that what somebody might have done is cut apart different parts of a gel, or maybe parts of two different gels, and stuck them together into a single image. Now, that could be negligence. That might be somebody not knowing that you're not supposed to do that, or that might be somebody purposefully messing with their data, altering their data. Say, removing bands or lanes from this gel, maybe that were in between there, that didn't show something that the authors wanted you to see. So the problem here with many things in research ethics is that if anybody has the slightest suspicion that you've done something wrong, that means you've done something unethical, regardless of whether you intended to or not. If somebody thinks that something unethical was done, that in itself creates a problem. So what our job is as scientists, understanding research ethics, is to avoid doing anything that anybody would perceive as inappropriate. So this is just one example. Elizabeth Beek is one of a number of scientists, but she's one of the most famous currently here in 2010 when this video is being recorded, scientists who make a hobby or career actually out of looking through published research manuscripts to try to spot image manipulation. And just to underscore the fact that this is a widespread problem, let's take a look at the abstract from this paper that she and co-authors published, where they looked at data in 20,621 papers from 40 different scientific journals, et cetera. And they found that 3.8%, almost 4% of those papers contained problematic figures. Doesn't mean that something was done wrong, but that something was done questionably. And again, the idea here is that something questionable was done. That calls into question the content, the results, the interpretation, the conclusions, and even the motivations of the individuals that are the authors on the paper. So there is a big issue with image manipulation in science. And so that's why there are rules that we want you to know about so that you follow them both this semester in Biology 104 as well as in the future. So at one point in time, at least a decade or more ago, the Journal of Cell Biology had this list of requirements, their policy on image manipulation. So I'm going to show you examples of all of these. And these were published by the Journal of Cell Biology, the examples I'm going to show you, when they published this policy. So you can't obliterate data. That's the first bullet point. Second bullet point relates to the gel image I just showed you, which is that if you want to combine different parts of an image, you have to do so explicitly. You have to graphically show that you're stitching together different parts of one image to make a new image. The third bullet point explains that if you're going to make a change, say if you want to apply a filter to an image, you can't apply a change to just part of the image data. You have to do the whole image, treat it the same way, or nothing at all. You can't preferentially select certain areas in an image to do something to, but not the rest of the image. And that's very similar to the bottom bullet point. The bottom bullet point also basically says something I'm going to ask you again later, but this is the first time you'll hear it, and you probably already know it. The best way not to get into trouble with image manipulation or any sort of data management is in the manuscript or in your lab report, just be explicit. Say exactly what you did. If you're transparent and you say, this is what we did, then you won't get in trouble. Somebody might say, well, that is a thing that you're not allowed to do, but at least it doesn't look like you've tried to hide it. That's worse. So here's an example of misrepresentation, that first bullet point. So on the left side here, we have the original image. So again, this was from the Journal of Cell Biology when they published that set of requirements. So this is one example they drew on to explain their policy and what to do, what not to do. So the original image that they got from the authors after this was caught is shown on the left, and the manipulated version that the authors of this, whatever manuscript this gel image came from, tried to publish. So what's changed? What's the difference between the two, and what's the motivation of the authors? Well, all of a sudden in this manipulated image, there's a set of bands in lane 3 that were not present in the original image. So what did the authors do? Copy, paste. And you can see that if you look, actually, I don't know, maybe it was that pair of bands or the next pair of bands, but if you zoom in, you see very similar pixel patterns between those two bands. And that's how they got caught, is that not just that those bands are the same molecular weight, but that this is actually a copy and paste. You cannot do that. That's inventing data. That's fraud. That will get you into serious trouble. So top version is the original. The bottom pair is, so this is the, actually, these are both original, excuse me. These are just from two different experiments that the authors are trying to publish like this. So the question is, what is the arrowhead trying to indicate here? These authors have done an experiment that they called, well, it was relabeled. This is experiment A and its control experiment. And here's a different experiment, experiment B and its control experiment. So what's the problem? These panels that contain the controls, that's okay, right? They've done well with the first bullet point because they've shown that they're not assembling multiple pictures into one image. They put boxes around them and a white space in between, which visually shows us that they're showing us two separate parts of a gel, and that's okay. What they shouldn't have done, which they did, was this panel, this image, this control experiment is exactly that image. In other words, they did one control experiment. They took this one picture, but now they're trying to pass it off as though they did that experiment again. That's data falsification. Even though this is a real experiment, if you run an experiment, you have to do the control experiment at the same time. So they did not do control experiment B at the same time they ran experiment B. They did this when they ran experiment A. So it's not a control experiment. There's no control experiment for experiment B in this case. And these authors got in trouble for trying to get this published. Now here's an example of what not to do again, related to our ability, especially with digital images, to change them in Photoshop or using filters on a camera app or wherever. We can see here that what authors tried to publish was this fourth version, where you see a single band that seems to be clean. There's no other bands in that lane. It's intense. That's this band, which in the original image looked like that. And even earlier, it looked like that. So how do the authors get from panel 1 to panel 4? Well, clearly what they're trying to show us, at least it's clear to me, I'll describe, is that this is a type of experiment where there are lots of additional bands in this lane on a gel. They just want us to see that the main band, the brightest band or the darkest band, the primary band is this band. And presumably that's a band of a particular molecular weight that maybe represents a protein that they're interested in studying. So what did they not like about their original image? Well, it's got this really smudgy band over here, and there are multiple bands in this lane, and they don't really want the readers of the paper to be distracted by, well, what's this band represent? What's that band represent? Maybe if we just display this really cleanly to the readers, like in panel 4, it'll be an easy interpretation by people that are reading the paper. They won't get distracted by all this other data. So what do? They increase the contrast, which means that anything gray either turns into white if it's light gray or black if it's dark gray. So if you adjust the contrast settings just right, you wind up with that lane, which just has that single band. And then they cropped image 3 to get rid of the other 3 lanes on this gel. So we get what they tried to publish was 4, right? This is not allowed because they're omitting data, they're covering up data. They're not showing us what the original data looked like. And this is a huge, huge gray area. there's a lot of debate over, well, what counts as obscuring data versus not. Because, for example, if they had taken this picture more quickly with the camera wouldn't have picked up these faint bands. The picture would have looked something like panel 3. And if you do an experiment that has a lot of different, if you run a gel, for example, that has a lot of different experiments on it, isn't it fine then to take a fast photograph? So the original would look something like panel 3, and then just to crop away the parts of the image that aren't related to what the authors are trying to show. So there's a huge gray area here about what's okay and what's not okay, and how to perform experiments and how to collect the data to ensure that you're not committing fraud, which this essentially would be based on the policies we've seen so far. Changing the contrast to omit background bands is fraudulent. Likewise, this is very similar. You're not allowed to change the contrast of one part of an image if you don't apply that to the whole image. On this, the good part about this example was they applied the change in contrast to the entire image. So every bit of that data was affected similarly by what the authors were doing in Photoshop or whatever program. But here, what the authors did was they didn't like the fact that that band was so dark. And maybe this is a dilution series, so the bands should go from dark to, if I knew how to spell light, L-I-G-H-T, in order. And somehow this band is darker than it should be. Maybe somebody screwed up and accidentally loaded those two dilutions in the wrong order or something, but they needed to lighten it up. So in Photoshop or some other program, they use the rectangular selection tool to select part of that gel image and just lighten that part so it fits with their expectation. That's fraud. That's both changing the data as well as only changing part of the entire image, which is not allowed. And I'll close with a few final examples of things that you might have done before or you might be tempted to do in the future. Here's an example of a nice looking pair of gels, or pair of bands, that is, 2 lanes in a gel. But there's this like unsightly dark spot there that has nothing, that's not data, that's some sort of artifact. But the authors don't want to publish it. So what do? They use Photoshop to paint over that with some background color. And they also get rid of these background bands. So they got rid of this artifact, which is not data. And they got rid of some of these background bands that are data. So how did they get caught? Well, if you zoom into the manipulated image, you'll see this repeatable series of dots. Right? That's not natural. That's indication that something happened. And so what they had done was they took a part of the background that includes that dot, and they pasted it over these bands to eliminate them with something that otherwise looks like just the background color of the gel. Not allowed to do that. If you get caught, you will get in serious trouble. There's one other point that I want to make, and maybe I mean to do this later in this presentation, but this is a good time to do it now. It's worth thinking about, why is it important not to remove background bands? So think about that for a minute. Why have we decided that it's bad to do this sort of thing? I mean, they haven't changed anything about these data. And if those are the data that the scientists want us to focus on, then what's so wrong with cleaning up the background? I mean, after all, they could have cropped the initial figure like that and just shown us that part, and then they wouldn't have had to deal with the background bands. So think about that for a minute. A couple other examples. So here's an original image with a super dark spot that doesn't represent data. That's, again, an artifact. And what they want us to focus on are these gray dots around these 2 ovals. I'm not sure what components of the cell that is, but they want us to focus on those dots that are surrounding, maybe that's a cell membrane or the membrane of some sort of organelle. So the authors in their manipulated image filled in with black those objects that they want to draw our attention to as the reader. Not allowed to do it. You're manipulating the actual data by doing it. And also deleting that really dark spot. It also, again, not allowed. Finally, although we've looked at this in the context of gel images already, here's another example, again, published in the same JCB paper, of something that looks like some tissue culture cells stained with some fluorescent dyes and photographed under a microscope. But when somebody opened this in Photoshop and they changed the contrast, they found that these two cells and this cell weren't part of the original image. If you change the contrast, you can see that they were cut out from some other part of the image and physically, digitally moved into the space that this group of cells already contained or already existed in. Presumably just to get more data into a smaller space, because especially back when this was published, it cost a lot of money to publish color pictures in print journals. Now that we've got digital journals, it's less of an issue, but there was a premium on space on the physical printed page. So it was better if you could get more data into a smaller space instead of showing more colored pictures. So presumably that's what motivates these authors to add in some cells probably from elsewhere in the same experiment. I mean, I'm not saying that this is necessarily a bad approach, except that you're not allowed to assemble multiple images into one field, because this picture is not an accurate representation of what the authors saw when they looked in the microscope. They only saw those cells in that area of the tissue culture plate, for example. So here, finally, we turn back to that Google Doc, please. So please, in just a minute, pause, go to our Google Doc, and provide some feedback on what you think are best practices for avoiding the perception of misconduct. Hint, we already talked about one way to do that in this video, so maybe just reiterate that to make sure that you remember it. And then what are some possible punishments for misconduct? And I'm going to add a third opportunity here based on something we talked about earlier, which is I'd also like to discuss why is it important not to, say, crop or obscure those background bands in gel images and pictures of fluorescent cells and others? Why is that so problematic? So we'll add that to the list. Why not clean up images? So please pause now, move over to our Google Doc, provide some feedback asynchronously, and then we'll conclude when you come back. See you soon. All right, welcome back. So to reiterate, let's finish up here. Best practices for avoiding the perception of misconduct. I'm just going to say best practice, and you may have come up with something else, and I'm not saying that that would be wrong, but I'm going to go back to be transparent. If you, in the research report, lab report, written report, say exactly what you did, and be clear about how you treated your data, be it data points on a graph or digital images, just say what you did. The worst that can happen is somebody will say, you can't do it that way, try again. If you try to sneak it past somebody and you get caught, the consequences are severe. Punishments for misconduct range from, for students, for example, failing the class, expulsion from school in Biology 104, which is maybe for you a required class, but won't be required in the near future. If you got kicked out of class, it's possible that the department would say you're not allowed to retake the class, which means that because it's a required class for the major, if you don't pass it, you don't get to graduate as a biology major, which means you have to switch to some other major, which means however many more years in school. Practicing scientists, when they're caught in misconduct, will get papers retracted, fines might be levied against them, they may be ineligible to receive any future funding, they could get fired from their position or not allowed to supervise people in research. There are a lot of different ways that this will go south, that is, go poorly for people that are involved in scientific misconduct. So lastly, before we turn for what to do this last week as an assignment, why not clean up images? What do you think? What are some reasons why it's not a good idea, even if there are those little faint background bands on the image or some other little speckles somewhere? Why is it not a good idea to clean that up or to crop it away? The main answer is that, especially for those background band examples that we've seen throughout today's video, we don't know what those represent. We think, maybe we, the authors, thinking from their perspective, might not know what they mean. They might just be a hassle, something that's going to distract the readers. Somebody else, though, someday might understand what those mean. They might be different splice variants of a gene that create different molecular weights of protein, or they might be proteins that have different covalent modifications, different chemicals attached to them, so they're different molecular weights. There are so many ways that in the future, other people might benefit from the data that we're trying to cover up to be clean looking. It's unfortunate. So that's the main reason not to clean up images. It's better to be truthful, to show exactly what the experiment looked like, and let other people be able to interpret those data in the future. That is the scientific process, and that will help you from getting into trouble with research ethics. So to conclude our exploration for research ethics in sort of active instruction this semester, thanks again, by the way, for having watched all of these and participating in our asynchronous Google Docs, and for all of the homework assignments as well. Here's the last homework assignment. And this is one that's based on work that I did as a graduate student. There's the before picture on the left, and I've got the after version that I created on the right. And this image on the right got published in a scientific research article. And during review, one of the reviewers caught me doing something they thought was scientifically unethical. Fortunately, we were able to explain what happened. I didn't do something unethical. Well, I sort of did, actually. We'll talk about this perhaps in class. But what I'd like you to do then, and the assignment that's due on Canvas for next week, is to compare the original image on the left and the final image on the right. Find all of the ways that I changed the original image to produce the final image, and then give me advice, just write out advice on, for each of those different changes that you spot, what should have been done better to make the final image a more ethical representation of the original data. So again, thanks very much for your participation. I hope you learned a lot that will be helpful, not just in this class, but in future classes and in your life as a scientist. I'll see you in class.